Good morning, and very welcome to the Royal Swedish Academy of Sciences. Welcome back, I should say, to many of you who were here yesterday. Today, we're celebrating chemistry. The Academy met in session this morning and took the decision on the Nobel Prize in Chemistry, and we have just spoken to the new Nobel laureates. I'm Göran Hansen, I'm the Secretary General of the Academy, and with me today is on my right, Professor Claes Gustafsson, who is the chairman of the Nobel Committee for Chemistry, and on my left, Professor Sara Snogerup Linse, who is a member of the committee, actually its previous chairman, and an expert in the field of this year's prize. And as usual, I'll read the announcement in Swedish and in English, and then the citation also in German, French, and Russian to cover all Alfred Nobel's favorite languages. Then we'll hear remarks about the prize from Claes and an introduction to the science behind the prize by Sara. This year's prize is about harnessing the power of evolution. Årets pris handlar om att bemästra evolutionens kraft. Kungliga Vetenskapsakademin har idag beslutat att utdela 2018 års Nobelpris i kemi med ena hälften till Frances H. Arnold för riktad evolution av enzymer och med andra hälften gemensamt till George P. Smith och Gregory P. Winter för fagdisplay av peptider och antikroppar. The Royal Swedish Academy of Sciences has today decided to award 2018 Nobel Prize in Chemistry with one half to Frances H. Arnold for the directed evolution of enzymes, and the other half jointly to George P. Smith and Gregory P. Winter for the phage display of peptides and antibodies. Zur einen Hälfte an Frances Arnold für die gerichtete Evolution von Enzymen und zur anderen Hälfte gemeinsam an George Smith und Gregory Winter für das Phagendisplay von Peptiden und Antikörpern. Pour moitié à Frances Arnold pour l'évolution du rejet des enzymes et pour l'autre moitié conjointement à George Smith et Gregory Winter pour le phage display des peptides et anticorps. Frances Arnold za naprawленную эволюцию ферментов, George Smith и Gregory Winter za fagový display peptidov и antitel. And on this slide, you can see some brief biographic data about our new Nobel laureates. Frances H. Arnold was born in Pittsburgh, Pennsylvania, in the United States in 1956, and she's currently at the California Institute of Technology, Caltech, in Pasadena, in the Los Angeles area. George P. Smith was born in 1941 in Norwalk, Connecticut. And he's currently at the University of Missouri in Columbia in the United States. Sir Gregory P. Winter was born in 1951 in Leicester in the United Kingdom, and he is at the Laboratory of Molecular Biology and the University of Cambridge in England. So with that, I'd like to ask Claes Gustafsson, chairman of the Nobel Committee, to make some remarks about the prize. Claes.
So thank you, Göran. So this year's prize in chemistry rewards a revolution based on evolution. Our laureates have applied the principles of Darwin in the test tubes and used this approach to develop new types of chemicals for the greatest benefit of humankind. In the natural world, evolution of proteins has been around for thousands of millions of years, all the way back until the start of life on Earth. Genes have mutated and proteins have evolved to improve the fitness of this life. For thousands of years, we humans have used selected breeding to create animals and plants with properties that have been useful for us. This year's Nobel laureates have taken the next step. They have used the molecular understanding that we today have of the evolutionary process and recreated the process in their laboratories, in their test tubes. In doing so, they have been able to make evolution many thousand times faster. And they have also been able to direct evolution to create proteins with new and useful properties. This work has led to the creation of proteins with new enzymatic activities, able to catalyze useful chemical reactions. In addition, with a method called phage display, they have also evolved proteins with new binding properties, such as antibodies, that can be used to treat disease. In their laboratories, our laureates this year have been able to direct evolution, to steer it, which has led to new chemical tools that can be used in everything from environmentally friendly detergents to the creation of new biofuels and pharmaceuticals.
Thank you, Claes. Sara, can you give us some insights into the evolution of enzymes and phages?
Okay, thank you. Life on Earth is the result of billions of years of evolution. Organisms have adapted their chemistry to fill niches and environments. Random mutations in their genes lead to changes in their proteins. Many of these are enzymes. Enzymes speed up the chemical reactions in biology. Any changes that are beneficial or do no harm may be kept for the next generation, for further randomization and testing during generations to come. Evolution has given us an amazing variety of creatures, full of little helpers, enzymes. Chemists use enzymes to speed up reactions in their laboratories, in industrial processes, and in consumer products. Over decades, scientists have tried to improve enzymes using rational thinking. Frances Arnold realized that to really get somewhere, she needed to grasp evolution and blend in an element of randomness into the process. In 1993, she published the first example where she had used directed evolution to improve the performance of an enzyme. She took the gene for the enzyme, introduced random combinations of point mutations, expressed all these genes in bacteria that were then producing a whole library of enzyme variants. These were screened for activity, and the best variants were kept for further rounds of mutagenesis and screening until a desired level of performance was obtained. To the left, there is a model of the enzyme that she has optimized in green. The blue is the substrate. As the diagram shows, the best variant after 3 generations of random mutagenesis and screening was 256 times better than the starting enzyme. In red, we see the side chains, the positions of the enzyme that were changed to achieve this. Nobody could have predicted that this particular combination would do the job. This illustrates the power of evolution and the element of randomness. Frances Arnold has since used directed evolution to improve a number of enzymes for new reaction conditions, to conduct new chemical reactions, to take in new substrate, even to make bonds for which no enzyme is available in nature. The applications are numerous and include molecules used for brain imaging, biofuels, pharmaceuticals. The enzymes used from directed evolution make us a greener chemical industry where they can replace more harsh chemical catalysts, even toxic metal ions. Enzymes derived by directed evolution are also used to make high energy molecules and reaction intermediates for synthesis. Now over to the other half of this year's Nobel Prize in Chemistry, where we award the phage display of peptides and antibodies. Now we get to meet a new little fellow, a bacteriophage. This is a virus that can infect bacteria. A bacteriophage is a protein capsule that encloses the DNA that codes for its own makeup. It fools the bacterium to produce new copies of the DNA and all the proteins and spit out a multitude of copies of the infecting phage. George Smith realized that this could be used for a powerful technology. He took a foreign gene, placed it inside the gene of one of the capsule proteins so that the encoded protein ended up on the surface of the bacteriophage. This we call phage display. In his first work, he displayed a fragment of a protein. This we call a peptide. And he mixed the displaying phages with a millionfold excess of other phages. And then he immobilized, in yellow here, the antibody that's specific for this peptide and used it to fish out the binding phages and wash out the rest. After this, he also introduced the concept of peptide libraries, where he displayed not a single peptide, but the whole library of different peptides on phages. And when he used the antibody to fish, he could then get the sequences with highest affinity for the antibody and determine its epitope. To really use the technology to derive therapeutic antibodies, the phage display has to be done in reverse. Gregory Winter took a fragment of an antibody, a fragment here in yellow that contains all the binding parts, and managed to display it in fully functional form at the surface of the phage. Again, he added a millionfold excess of other phages, and he could use the target of the antibody as a fishing hook to pull it out and remove the non-binding ones. He then went on to make libraries of antibodies on phages, and using an immobilized target, he could add a library, wash away the weak binding antibodies, extract the high affinity ones, and subject them to new rounds of diversification and screening to select better and better antibodies. And over just a few generations, he could obtain very high affinity and very specific antibodies. And this forms the basis for a pharmaceutical revolution. Antibodies produced by phage display are used to treat diseases such as autoinflammatory diseases, anthrax, and cancer, and many more are currently in clinical trials. The methods developed by Francis Arnold, George Smith, and Gregory Winter are to the highest benefit of humankind. Thank you.
Thank you, Sara. I think we may now have one of our new Nobel laureates with us on the phone. Dr. Arnold, are you there?
I am there.
Hello again. This is Göran Hansen. I'm the guy who woke you up in the middle of the night less than an hour ago. Have you had your cup of coffee yet?
Not quite yet. Had to splash water on my face.
So we are sitting now in the session hall of the Academy of Sciences, and here are nearly 100 journalists in total, and I'm sure some of them would like to ask you some questions. So are you ready for them?
I am.
Shall we give Thomas von Heyen a chance to start?
Thank you. Congratulations on the Nobel Prize.
Thank you so much.
We have a paper here with a headline that says "Playing with Evolution," and I was thinking maybe something similar has been said about what you do earlier, and sometimes a phrase like that could make you suspicious. What are you up to? What are you doing? Have you had lots of questions like that, and what do you usually say?
I do get questions like that because we're curious.
Now we have some problem with the line, I'm afraid. Sorry to interrupt, but we cannot really hear you, Dr. Arnold. Let's see if we can do something about it.
Thank you.
Okay, let's try again. So please go ahead.
I have encountered that it's still a problem.
Okay, what a pity. The line doesn't work. So much for sophisticated electronics. I wish they were evolutionary developed enzymes instead. Yeah, no, I don't think we get anywhere. Want to give it another try? We can give it one more chance. No. They have to save their questions for when you come here in December. Thank you so much for trying to be with us during the press conference, and we look forward to meeting you in December when you come to receive your Nobel Prize. Thank you and bye-bye. Well, I'm afraid you'll have to do with us in the panel instead. The lady over there. Could you wait till you get the microphone?
Sorry, could I stand up or just sit here?
Just sit.
Okay, thank
Please.
you. This is Janet Wang from Nordic Chinese Times, and we noticed that many of the Nobel Prizes in physics or chemistry during the decades have been awarded to the findings related to cancer, biology, or physiology these years. And so my question is, could I ask if why are these phenomena increasing these years, and what would the Nobel Prize Committee suggest in the future? Thank you.
I think we'll ask Claes Gustafsson to comment on that.
So there is no sort of real intention from our side to sort of focus on these subjects that you bring up. Could be true if you go back to that there could be some pattern there. I think it has to do with the fact that, you know, Alfred Nobel, he wanted a prize that was for the greatest benefit of humankind. So it had to have consequences for people, for humans. And I think that because of we're always looking at both brilliant science, but also brilliant science with sort of consequences. And maybe that's why you can see that some of the prizes are related to the area which you allude to.
Thank you so much. I think these findings are really inspiring and marvelous. Thank you.
Good to hear. Good to have you with us. More questions? Yes, the gentleman from Associated Press.
Thank you. So I understand that you spoke with the laureates. Would you be able to tell us a little bit how they reacted? Have you managed to speak to all of them, and how was their initial reaction when you told them the good news?
Yes, we spoke to all three of them, and they were all very happy and enthusiastic and exhilarated. They look forward to coming here in December, and they were also happy to share the prize with their colleagues. So it was all positive reactions. We spoke with Greg Winter first, and then with Francis Arnold, and then last with George Smith, and they were all delighted. Yes, please, Per Snapret from Dagens Nyheter.
I wonder if there are any patents in this field that have economic significance for pharmaceuticals or other products.
Sara, would you like to answer?
Yes, Francis Arnold holds a whole range of patents. Enzymes that make biofuels, for example. So you can now make fossil-free fuels for cars and airplanes based on her technology.
From what? From what substances?
From different grown substances. So it's not taken out of the fossil reserves. So renewable resources. I think she holds something like 10 to 15 patents. I am not aware of the exact number. Gregory Winter also holds patents for some of the antibodies that are derived by the phage display technology.
Mm-hmm. Thank you. More questions? This is your chance. Yes, please.
Thank you. I'm Sufichan Aksasong from Green Post and China Radio. I'm sorry, I like to ask you to use simple words to explain short how significant their discoveries are.
I'd say very significant. But I'll give Sara a chance to say something more.
Yes, if we start with the first half, the significance, one significant is that you can now use enzymes to speed up, in principle, any reaction you want using these protocols. And you can derive enzymes that replace previous toxic catalysts in industrial processes. So I think it has a very huge significance. And the second half makes it possible to derive fully human antibodies using the phase display. These libraries are now composed of fully humanized antibodies from the adapted, and they can be made with very high affinity and specificity so that you can use very small amounts in treatment.
So perhaps if I add safer, greener chemistry and antibody drugs with better efficiency and less side effects. More questions? Yes, please, gentlemen over there.
Are there any potential damages in these researches, like such as create super species could be damage humankind?
I think it's
Claes.
Should I answer? So there are, of course, with all science, there are, of course, risks. But when you think about the science that we talk about here, it's sort of heavily regulated by, it's gene-modified microorganisms and sort of are used all over the world in different labs. And we use them today to produce things like insulin, for example, treating millions and millions of people. So this is that there is a rule-based system that is sort of well established and that regulates this work also. Then of course evil minds can always do things with technology, but I don't see any particular dangers with this price compared to many other things.
More questions? If not, thank you very much, and we'll close this press conference, and there will be individual interviews. Thank you very much.
Yes, absolutely.
Go towards the corner.
Thank you.
Sara Snogerup Linse. This year's prize is for engineering proteins. Can you tell us what are proteins?
Yes, I like that question. Proteins are molecules, rather large molecules that you find everywhere in your body. They conduct almost all the processes that go on, the memory, the thinking, the coagulation, the muscles. They are really the workhorses in our bodies.
So if they are so plenty, why to produce even more?
If they are so plenty.
Why do you need to produce even more?
Yeah, because in the body, some of the proteins we have or other organisms have are enzymes that speed up chemical reactions. But there are reactions we want to do in chemical synthesis or to produce fuels that those enzymes can't do, so we have to improve them.
This is about enzymes produced by Frances Arnold this year.
Yes, that's the one half of the prize, and that is to make the enzymes conduct new chemical reactions or better the reactions they do today. And the other half is about improving binding proteins. So these are proteins that attach to something and conduct biological processes by this means, for example, antibodies. And this technique can then be used to produce human antibodies against pathogens or other substances.
And to come back to the main method in both prizes, so to say, used, you were talking about evolution. And when we think about natural evolution, you think it's often described as a combination of chance and necessity. Is this what they did?
Yes, so this is, I would say, a combination of chance and necessity, but also quite a big element of thinking, because what they have done is to really speed up the evolution. Because nature has had billions of years, but now you want the process to be possible in maybe a few weeks or a year or something in the laboratory.
So it was like random experiments and then picking up the right parts of it?
Yes, I would say in nature it's a rather random process because the mutations happen by chance, maybe due to UV radiation or something. Whereas in the laboratory it was a combination of knowledge base. So you have to have some idea of which positions to vary, but then you add a randomness on top of that.
So the biggest difference would be that you know what goal you have.
Yes.
While in nature, actually there is maybe no goal.
Yes, I would say nature has no goal, but what works, and everything that makes things better or that you can populate a new niche is then kept. Whereas in the directed evolution, the scientist defines the goal. I want to break this bond or make this bond, and I want to make it as quickly as possible at 58 degrees or whatever is the condition. Yeah, so the scientists define the condition.
And one of the prize laureates, Frances Arnold, she knew she wanted enzymes. Why couldn't she design them from the beginning?
It's a very good question, and scientists also try to do that. And in principle, we know which kind of intermolecular forces operate in proteins, but I think the problem is that it's a multi-combinatorial problem, so it's still not possible with the knowledge-based methods today to design an enzyme from scratch. One can design enzymes that have some catalytic efficiency, but to really, really get somewhere, you have to bring in the power of evolution to optimize them.
Can you tell us something about who is Frances Arnold?
I think she is a very good and she's a very talented scientist, a very curious scientist, and also goal-oriented. She knows what she wants to do, and then she sets out to do it.
And she made it.
She made it, yes.
Also, the other prize laureates, George Smith, he designed a method with this phage display, which is really hard to understand what it is, but it's a method to produce antibodies.
Yes, it's actually a method. So the way he designed the method, he took those phages that are these protein capsids that have the DNA inside that codes for making the capsid. And what he did, he put in the gene for the protein he wanted to study, and he then sort of fooled the phage to put it on its surface. So this is what we call phage display, on the top or on the bottom, depending on how you turn them around, but at the tip of the phage. Yes, and I think the key element of his technology that really made it fly was this physical linkage between the gene and the protein product, so that you could fish out phages and then take out the DNA and work further with new generations of variations.
And then Gregory Winter used it to produce antibodies. What is special about antibodies?
The special thing with antibodies, I mean, it's a part of our immune defense. These are proteins that nature have designed to have a possibility to become very specific. So the phage technology harnesses that power. Antibodies have 6 different loops in each binding pocket that it can vary. And this is ideal, in fact, to put on a phage and do random mutagenesis to search for very high affinity antibodies, so they can have very high affinity and high specificity for a certain molecule and not react with others.
And then they can be used to make drugs, actually.
They can be used as drugs because they can either bind to objects or surfaces we have in the body, or they can bind to things that would try to invade us.
How many drugs have been produced from this method?
I actually don't know, but I know that of the 15 most sold drugs on Earth, 11 are made by this method.
Are they very expensive?
I'm sure they are very expensive, but the advantage with the phage display is that you can derive antibodies with such high affinity, much higher than you get with immunization, that you can lower the doses. You can use much lower doses, you can even use them cutaneous on the skin, you don't have to inject them. So there are many advantages with them.
So what do you think is next? What is the vision for the future?
The vision for the future, I think we're just at the start. I think we will see many more enzymes that catalyze bonds that we had no clue that an enzyme could actually do. One of the latest examples from Arnold's lab are enzymes that catalyze the formation of carbon-silicon bonds. And these are bonds you don't find in nature, and there is no natural enzyme, but she managed to take one and evolve it to form these kinds of bonds that are important for making materials, for example.
New materials.
New materials, yes.
Thank you very much, Sarah Snodilo, for being here with us.
Thank you.